I'd like to welcome our panelists. You recognize, first of all, Jules and I will be helping manage the Q&A and the chat. So if you have any questions or you have any technical difficulties, let Jules know. I'm substituting for Sarah, and she's done, dozens of these. So I will do my best to channel her today because she's an awesome facilitator. But she is going camping with her two. Her 6-year-old, her 3-year-old, and her husband. She's braver than I was when my kids were at that age. So they're off camping today. So I'm covering for her. I'd like to introduce Dr. Tom Klak. He's the Vice President and Chair of Gene Conservation in our main chapter. He's a professor at New England University at Bedford, Maine. Longtime enthusiastic volunteer and helping us speed up the science with the transgenic program. Welcome, Tom. Also Dakota Matthews. He is the molecular lab manager at the College of Environmental Science and Forestry at SUNY and with the American Research Project. He's currently in Lisbon, Portugal, and bombing around Europe, evidently, until he comes home tomorrow, is it?
I fly back Sunday morning at 5:40, so I'll be in America at 6 p.m. It's a 20-hour flight.
Well, have fun with that.
Thanks.
And to the participants, I see lots of familiar faces. Thank you so much. Last but not least, we have our own TACF Lily Kingsolver. She's our nursery manager at Meadowview Research Farms. She's been with us for over a year and a half now, and she's going to show us a really cool new facility that we built. And we put in all kinds of highlight work in there so we can get the pollen to respond quickly. And Lily's going to share some photos about that. So I'm really excited about today's Chestnut Chat. And with that, Jules and I and Tom and Lily will go dark, and we'll tee up Dakota to go first with his presentation. Any questions before we get started on the technicalities of Chestnut Chat? Nothing right now. Everybody, if you can see us, if you can hear us, we're good. And throw questions in the Q&A and throw comments in the chat. With that, Dakota, take it away.
Thank you, Lisa. Sorry. Hello, everyone. My name is Dakota Matthews. Like Lisa said, I'm the molecular lab manager at SUNY ESF. Working with Dr. William Powell. I got my master's with him back in like 2019. And I've been working there ever since, enjoying every minute. And today I want to talk about transgenic American chestnut pollen production, basically how it started and where we're at now. And then that'll lead into Tom's research and the American Chestnut Foundation's new building that they've built that Lily's going to show you as well. And then just quick right here, this is a SEM image of chestnut pollen embedded on the outside of an anther. I just really like the picture, so I thought I'd throw it up there. So yeah, let's get started. So the initial development was actually a scientific accident, like Lisa was talking about earlier. It was developed by Kathleen Baer over here on the right. She was actually the one who taught me everything. She was a wonderful human being. And basically, we wanted to accelerate the growth and maturation of Chinese chestnuts for hybridization studies, as well as breeding, and just an assortment of assays that we wanted to do that we didn't want to wait for them to mature in the field over many growing seasons. And so we had this old school highlight growth chamber you see over here on the right, and we started placing Chinese chestnut in there, and lo and behold, around 8 months later, we noticed that the Chinese chestnut started producing catkins, and as well, sometimes they produce female flowers. So you can see the burrs over here. And we didn't intend for this to happen, but the implications were awesome because it had the prospective ability to accelerate our breeding processes much faster than the normal 6 to 8-ish years for a chestnut tree to mature in the field before it's ready to either collect pollen or pollinate female flowers. And actually, this is something that you can check out in the archive. I know Jules posted it in the chat of the Journal of the American Chestnut. This was the issue from the May-June 2012. And so this is when we were first starting to develop this. So initially we did this with Chinese and American or Chinese chestnuts, but quickly we started to put in our tissue culture generated transgenic American chestnuts in the hopes of producing pollen to accelerate our crossing to produce genetic diversity, as well as start to expand our breeding operations. So our current protocol for pollen production, starting from that accident, is we have our transgenic American chestnuts, or chestnuts from our transgenic American crosses. We don't know which one is which because there's a 50-50 chance of inheritance. So we need to do a histochemical test to assess which chestnuts inherited the transgene oxalate oxidase and which ones didn't. So you can see in the picture over here on the left, we take a core from the endosperm tissue of the chestnut right after harvest. And you can see here, we take them from the left and the right side. We usually only do one, but in this case, we did two. The chestnut embryo is up here in the top. So we need to make sure we avoid that so we don't accidentally damage the embryo and ruin germination. So we collect these chestnut cores and we place them into histochemical assay solutions. And basically, in the presence of oxalate oxidase, the American chestnut endosperm tissue will turn this very stark black color, and that would represent a transgenic American chestnut cross. And then there's a negative solution right here to prove that it's not something like extra hydrogen peroxide within the nut tissue that would trigger the black colorations. If this was a negative chestnut with no oxalate oxidase, both of the nut cores would appear as this just white plain tissue. So once we've isolated which chestnuts are transgenic and which ones aren't, we put them in, we label them individually in tea bags and put them into cold stratification for 3 months approximately. And during that time, we coordinate with all of our collaborators and try to decide which crosses that were successful and transgenic from the previous year would be best to initiate into our breeding program and produce pollen for further generations. This is to increase diversity as well as unique traits. And after that 3-month cold stratification, we pot them in the normal greenhouse for initial germination so we don't stress them out too much with the high intensity light, and get them to a relatively decent size. We usually leave them in there for about 6 weeks, and then we transfer them to the highlight growth chamber, which you saw in the other image. And pollen production is usually anywhere from 6 to 11 months. So here is the our first image of a transgenic American chestnut just beginning to produce a catkin. In this growth chamber, the humidity and the light are strictly controlled, as well as the temperature. We put them on a 16-hour photo period, which mimics peak growing season when they would normally be flowering anyways. But these trees are only, anywhere from 3 to 6 months old at this point. They're not developmentally ready to flower on their own. So tricking them into thinking that they're an old tree, we just keep them under this light constantly. And this kind of creates some developmental weird growth patterns in some of the trees. You can see the inner nodes in between each bud get really short over time. So you'll notice throughout these pictures here that, this spacing right here will shorten and shorten and shorten as it's consistently exposed to the 16-hour photo period. And then we eventually start to see these emerging catkins, and they eventually start to get bigger and bigger. and they flush right out and start to look like normal catkins, but they don't produce the catkins the same way because, again, these trees aren't normally doing this. So we're stressing them out. We are treating them poorly. We treat them as martyrs. They're going down for the cause and producing pollen for us. But they do produce very nice catkins, even though the trees look pretty crappy. That is the whole point. So as you can see, they kind of grow this bushy pattern where the leaves are intermixed with the catkins. They're really dense. There's no internode spacing anymore because they're just peak growing, putting out leaves as fast as possible. And then, so here's the very short internode spacing that I was just talking about. This is very common. And trees of the same genotype, the same crosses, if we don't put them in this treatment, they don't act like this. So it's not a function of the trees that we're actually breeding. It's just the treatment of them that make them grow like this. And they eventually start to flush out their catkins. So these are the anthers and the stamens. So the original image that I showed on my title slide, if you were to zoom in onto one of these little yellow balls, that's what you would see, those little brain-like structures with the American chestnut pollen adhered to the outside and the inside. So right about this stage is when we really start collecting pollen in earnest. We collect them on glass slides, which I will show you in a bit. And the static cling of the glass slide is actually a great vehicle for chestnut pollen. It's much more elegant than our original method, which was collecting in glass jars and using razor blades to scrape it all up and drop it in a jar. A lot of pollen was wasted, and we couldn't be as precise when we were applying the pollen to the chestnut flowers. And so I tried to get pictures throughout their pollen production cycle. And as you can see, the trees progressively get worse and worse looking. And the worse they look, the more pollen they produce. Because when they're stressed out, first thing they want to do is sexually reproduce before they die. So with specific trees, sometimes you can get multiple cycles in. So they will produce these pollen, these catkins, produce pollen. They will drop all of their leaves, dehisce their catkins. As you can see, they're dying on some of these inside ones. And then they'll start producing a whole new batch, and they look even weirder now. The chestnut leaves sometimes don't even reach full maturity. They grow in this weird gnarled pattern. But they produce even more catkins. And this is exactly what we want them for. I included this one because something we kind of also discovered by accident is that once we initiate catkin formation in these trees with this highlight treatment of a 16-hour photo period, we can move them to the greenhouse and they will continue to produce pollen on the catkins that they initially started in that highlight treatment. So this is a good way to save very important plants if they're not doing so hot and they're already producing catkins, but we want them to last a little bit longer. The reason that these are out of the highlight growth chamber currently is our highlight growth chamber broke. They're very temperamental and very old. So luckily we were able to induce pollen production this year on this batch before our chamber broke. So right now we're actually collecting and producing all of our pollen in a normal greenhouse under a normal, no added light, just natural light, and they're producing just fine. Looking a little bit better actually. And then just another thing, anecdotally, some trees just don't make it. It's not specific. We usually just have a couple of trees out of a batch of 20 that end up aborting. Like I said, this is a very harsh treatment. The trees are deciduous. They are meant to have dormant cycles. We don't let them. So some trees just don't do well. And this is a good example. We had 2 this year of our 20 that no matter what we did, we just couldn't save them. And they ended up just giving up. So that's a natural part of the process. And that's why we always have duplicates of all of our trees and crosses for specific ones that we want. So briefly, our collection and storage method, like I said earlier, we rub the glass slides along those catkins. The pollen is statically clung to the glass, and we place them into these glass slide jars that are used for cancer research, actually. And they store 4 slides in a jar, and then we place them in a desiccation chamber overnight for 16 to 24 hours. Doesn't really matter as long as it's within that range. And then we're able to store the pollen in the freezer and use it for our pollination season if this pollen was produced not during our pollination season. Over here is just another image of chestnut pollen up close. And then briefly, pollen preparation for the field. We rehydrate our glass slides in the slide jars in a humid chamber the night before. So that's just a like a shoebox basically with wet paper towels, and then we crack the lids, let them rehydrate in the refrigerator overnight, and then we store them on ice while we're pollinating in the field. The right here, you can see the American chestnut pollen is just coating these slides. It's a very elegant way of applying the pollen and not wasting it. You can see, if you look closely, you can see the pollen collected on the ends of the stigmas here, or the styles. And whoops. And the nice part about these slides too is you can actually see the track marks of where you have previously pollinated with. So it's actually very easy to see, oh, I'm running out of pollen, I might need to switch. Or this is still a good slide, we can keep going with this. So that is all of our pollen currently that we're using right now has been stored in the freezer using that method that I just showed you from the highlight growth chamber. And then right here on the left is another SEM image, and this is a dehisced anther head. So the outside of my initial image, this is the inside. So it's coated with chestnut pollen, and this is what we apply to these glass slides. So all of that is statically clung to the edge of the slide and is very easy to scrape off onto the American chestnut female flowers for our controlled pollinations. And with that, that is a brief intro to our pollen production, collection, and pollination process here at ESF. And Tom and Lily will take it from here, showing how they've further continued with this science. Thank you.
Thank you, Dakota. That was awesome. I love the creativity behind science because it's just really cool how you problem solve through creativity, which is a happy combination of analytics and creativity. Just so you all know, a few questions are bubbling up in the Q&A, but we're going to wait for Tom and Lily to finish before we start managing the Q&A, but keep them coming if you need to. And Tom, all yours. Thanks again, Dakota.
Okay, great. Everything on the screen there like it should be, Lisa? First slide.
Yes, I don't know if you could see me.
Okay, great. Okay, great. All right, so thanks for this opportunity to share the science of pollen production. I'm at the University of New England and part of the main chapter of the American Chestnut Foundation. So this slide, I think, kind of summarizes what we do. On one hand, we're pollinating wild trees out in the field, like this big 50-footer. In fact, I'll be up putting controlled pollination bags on this tree on the left later today after we get done. And then on the right side, we're getting the transgenic pollen production out of high intensity production here on the right. So it's a combination here that is what we're working on to bring the transgenic crosses forward. So in another way to sort of portray what we're up to, I'd like to talk about the two sides of the same chestnut burr. So on the left side, spend a lot of time finding, propagating, and pollinating American chestnut trees. That's the nature's genetic diversity that's so crucial to restoration. And one of the major ways we do that is through germplasm conservation orchards, or what we abbreviate GCOs. And in Maine, these GCOs, the oldest ones, are now get into their 5th year, and they're beginning to produce female flowers in some numbers, not major numbers like a wild tree would, but some numbers. So that's becoming really important. So that's the natural genetic diversity, so crucial to the project. And then the other side is the production of the transgenic blight tolerant pollen. And that goes on in labs as Dakota described, but also happening here at the University of New England. So what I'm gonna, then so it's the mom's on the left side and the dad's on the right side, put it that way, even though it's a bisexual tree. We match them up that way. So what I have to say, or what I know about producing pollen, comes from all we learned at ESF in Syracuse, Environmental Science and Forestry College, where Dakota is. So visits to there by people from New England, including myself here on the left, are how we learned the ropes and basically learned everything we could from there, and then tried to practice our own work here up in New England. And
excuse me, we have a raised hand. Brian Roth, the famous Brian Roth. Can you throw your question in the Q&A? Because everybody's muted. Okay, we have two participants raising their hands. Sorry to interrupt you.
No problem. Yeah, great. Now go ahead and interrupt as much as needed there, Lisa, no problem.
Oh, Brian Roth said, carry on, my bad. No problem, Brian.
Okay.
We're all challenged with technology. And John Venko, if you could hold your question till the end, that'd be great. Thanks, guys.
Okay, so the point here is that ESF is the base of everything we do, and we're kind of an annex up here at University of New England in Maine, learning from them and carrying on. Okay, so our production here in Maine started about 2 years ago when we brought home these 12 transgenic seedlings here on the right side, that very, unassuming, not very significant, variety of different crosses from ESF. And we tried to do our own thing here. I wasn't very confident. Here you see Doug McLean being much more confident as usual than I am, trying different systems of trying to produce pollen indoors. And so by then, that was July of 2019. By November of 2019, the first of those 12 produced catkins. Here they are. And then by later in November, we had pollen off of those same catkins. And it takes 3 to 4 weeks from catkin development to pollen development. And it's red because the LED lights are red. So so that's how it all started here. We're very excited about that moment. I'm sure Lily's looking forward to that as well in terms of her setup there at Meadowview. We have a growth chamber that we use, similar to what Dakota described for ESF, and get pollen that looks like this. Very interesting. Dakota was showing the electron microscope pictures. It's just amazing that pollen is so tiny. It's only 15 to 20 micrometers in diameter, and a micrometer is 1/10,000th of a centimeter. So to put that into perspective, it's incredibly small to produce a giant tree. So you can get a lot of pollen off of a seedling indoors under the high intensity conditions Dakota described. Here's one that came out of our growth chamber, and that's a lot of pollen that sticks to our slides. Here's another one, another seedling that was produced solely within our greenhouse. So we didn't use our growth chamber at all to produce this one. We were trying to see whether just a non really super expensive tens of $1,000s machine like the growth chamber, could you do it elsewhere? Could you do it under LED lights? And this one is an example that it is possible. And I think Lily's operation is replicating and enhancing this idea there at Meadowview. So what are the methods? Dakota mentioned, basically we follow very similar kinds of things, 16 hours a day, daylight or highlight, both blue and red light. Temperatures, we regulate from the high 70s to the low 70s in the day, high 50s at night. Relative humidity, kind of in the middle. pH needs to be lower than what comes out of the tap, so we have to adjust for that. We bombard the seedlings with fertilizer, all kinds of different types of fertilizer we mix in. For us, that helps to bring in different kinds. The pots we like to use after experimentation are 2-gallon pots. And the biggest issue is of all, and Dakota had mentioned this, maybe he has less of a problem than we do, are the various pests and pathogens. That's the crucial conundrum for producing pollen. It's a zero-sum game, whether the pests are winning or whether our pollen production is happening. So we collect the pollen on slides off of the seedlings, as you can see in this picture. We put it on the slides you see there on the top, and we group them into clusters of 4 because 4 slides can fit within each one of these pollen vials that get stored in a negative 80 Celsius freezer until we need it. It looks like that. Here's one of our freezers here in the bottom left. And then we send these pollen vials with 4 slides in each vial out to all our collaborators throughout the native range. And this is how we're developing our crosses and our genetic diversity of on the one hand, transgenic pollen, and on the other hand, wild trees. And we just did that on Monday, both from ESF and from my university, UNE. And we sent it out to about 9 or 10 different collaborators throughout Eastern US. Very exciting. Very interesting distribution of pollen production per seedling. I want to burden you with too many graphs, but this is basically a layout of the 24 seedlings that we started with here at UNE. And what it turns out to be is that there have been 2 seedlings that have produced a little over half of all of the pollen slides that we've produced. A pollen slide can pollinate about 15 female flowers in the field. So these 2, and this is a slide we produced a couple months ago, have produced over 500 slides each, whereas a few other ones have produced some pollen, but not very much. And the majority of the ones we started with hadn't produced any at all. So it's very skewed in favor of certain seedlings that are strong enough to handle the intensity and also to battle off the pests and pathogens. Very, very uneven distribution in terms of which seedlings produce. And then we test them to see if they have tubes. Tubes are what the pollen grain uses to reach the female egg in the flower. And so see these tubes off of the pollen grains. We're very happy, and that indicates that our pollen is viable, and we keep track of all of this. Here's another picture like this. This is under just 100 times magnification microscope with a camera attached to it. And when we see these tubes, then we're very happy with the success of our pollen production. And then we take it out into the field, and eventually in July here in Maine, and we put the pollen on wild trees, like you see here. We get a couple people up in the lift on the left and a person on the ground. That's actually the president of the Maine chapter, Al Faust. He's keeping track of the data, which pollen we're using in which cross. And we got all that data very well documented to know which crosses go where. And here we are putting the slides with the pollen on female flowers in the field. This is one of my the graduates from my environmental science department working with me last summer. Here's a little bit closer up look at pollinating using the slides from the lab. And just a real brief comment about the pests and pathogens, because for us, this is the big challenge of producing pollen over time, in the long term, over a year or 2 indoors. What happens? We get all kinds of aphids, green aphids and white aphids. They look like that, and you got to battle them. Each one is a different kind of a problem that requires a different kind of a solution, constrained by what you can do indoors within a university setting. So you can't use all kinds of different potential pesticides that you might come up with in an indoor setting like that. Very strict. Spider mites are another problem. Here's what they look like. And they erupt very quickly. So like from one day, things are all hunky-dory, and the next day you've got a big problem. Mealy bugs, another one. Different kind of treatment required. Powdery mildew fungus. All of these things can kill the plants. And so it's just a lot of effort to try to figure out what we can do to try to keep our seedlings happy. Fungal gnats, big problem. We battle them, we catch them on sticky sheets, yet there's still more of them. So that's big. And what happens then with all these pests and pathogens is they produce a pattern like you see in this graph. So this is just an example seedlings production of of pollen over time. So over on the y-axis on the left, you have how much pollen is it producing per day. And across the x-axis is the number of days since we planted the seedling. And so you see, basically, typically what you find is that it takes a long time, almost a year, 9 months it could take before the seedling is big enough and strong enough to produce any pollen. And then it can peak. And you see, we can get a lot of pollen there for, say, a week or two. And then the pests and pathogens kick in, and you see that kind of roller coaster pattern. And so it's that kind of a zero-sum game between production of pollen and pests and pathogens. We figure out what to deal with the particular aphids, if it's that, or the mealybugs, and then we can get some more pollen. So it's that kind of a roller coaster up and down pattern over time in our experience. Okay, so what we've begun to do then in our germplasm conservation orchards, DCOs, is find saplings that are precocious and are beginning to produce female flowers early. Here's one that was only 4 years old, and we're beginning to put the pollen on them. Now we're starting to get more genetic diversity from saplings in our GCOs that are representing all of Maine, all really different parts of New England and even beyond. And another one cool thing that we've done this year as an experiment is we're trying to see how fast can we produce pollen, and can we produce pollen within the same year as we collected it. So this is a seedling that we started in December of this year. It was a seedling that was initially created through pollination last July, in July of 2020. And so that seedling then on the left produced this catkin last month by early May. Okay, and then that's what the plant looked like by early May. And then by early June, early part of this month, it had already produced pollen. So that was the first time we've been able to produce pollen within the same year and advance the genetic diversity of the transgenic seedlings an entire generation within the same calendar year. So we can take this pollen then on the right and apply it to trees, and we hope we'll be able to replicate this and learn from the mistakes that we made, like pushing the light too hard early on, as Dakota mentioned. That's something you want to avoid. But it was a breakthrough for us that we were able to produce pollen within the same year that it was originally fertilized. And then lastly, where does all this go? Well, all of these transgenic crosses that we produce in Maine, as well as representation from the other sites throughout the native range where we sent our pollen last year, are out here in what we call a common garden experiment. And there's 550 seedlings planted here in Cape Elizabeth, Maine. Not only the transgenic crosses, but also a whole variety of different controls. They're full sibling non-transgenics that didn't inherit the gene. We have hybrids in there. We have Chinese chestnuts in there, F1 50-50, Chinese American chestnuts. We have the whole range, to see how they do in comparison and see how they perform in the field over time. So that's sort of the end result in terms of where the science is as we make our transgenic pollen in the lab and apply it in the field. So that's a whirlwind view of what's going on here in Maine. I'm going to turn it over to Lily to talk about things going on at Meadowview.
Thank you, Tom. That was great. And Lily, I can't wait to have you show off this cute little building. I know it's a scientific facility, but it's awfully cute too. Tom, that was so inspiring how much progress you've made. I'm just excited to hear about it. So thank you. Lily, take it away.
Thanks so much. Thanks to Dakota and Dr. Klak. They did great presentations. So as Lisa said, my name is Lily Kingsolver. I'm the nursery manager at the Glen C. Price Research Farm in Meadowview, Virginia. I want to start with a disclaimer, which is that everything I know about speed breeding, I learned from Dakota and Dr. Klak. So I don't have a ton of insight, a ton of new information, but I am really proud of the setup that we've just completed this spring. So I want to just show you a few pictures and kind of explain what we're hoping to do. We don't have any idea yet whether we'll be successful, when we'll be successful, but we're very hopeful. So I guess Dakota kind of gave you the past, right? And Dr. Klak is currently producing a lot of pollen, so that makes us the ghost of speed breeding future. I also want to say a really quick thanks to our Director of Land Management and one of our research technicians, Dan McKinnon and Jim Tolton for working so hard on the planning and construction of this building. They've just been hugely instrumental and very kind to me when I knew nothing about construction. So thanks to them. Now on our next slide, thanks to Dr. Klak for running my slides too. This is the interior of our building. When we started considering speed breeding, we had to consider, you know, what we're going to do, what kind of facility we wanted to start these trees in. And when we looked into highlight growth chambers, like the kind that Dakota showed you pictures of, they're extremely expensive. Not shocking, right? They're very specialized, very expensive. So we started thinking about how could we replicate the conditions within a highlight growth chamber by ourselves. You know, how could we do this by ourselves? And when we weighed all the different options and all the different costs against how many trees we could have in each situation, it turned out that getting a prefab shed from Home Depot and putting all this stuff, like retrofitting it with all this stuff, was the most cost-effective option for the number of trees we could fit in it. So this is just a shed from Home Depot, totally made of particle board. We insulated the inside. At the top there, you can see our air duct. That's just an outlet fan with a sensor. It kicks on if it goes above 80 degrees in there, because the lights do keep it kind of warm. Our oscillating fan there makes the trees, moves them back and forth a little bit, which helps with lignin production, makes them stronger as they grow. So that's why we've got that fan going all the time. So we researched this for about a year before we started building it. And then in February, we started purchasing materials, started construction in March, and finished this in April. So these trees have only been in here for about a month and a half. Someday we could have up to 150 trees. We have the capacity for that, but right now we only have 59 because we're just trying it out. Like I keep saying, who knows what's going to happen? But we're really excited about the potential. It seems like we're headed the right direction. So you can see these trees are sitting on plastic trays. Those collect the water. The floor is coated in hydrophobic paint, but we still try to keep the water as controlled as possible. And we chose not to put an outlet drain, but instead to shock back out the water every day because, like Dr. Klak was mentioning, pests and pathogens are a huge problem, and every hole you put in the building is a potential, entrance for pests. So that's why we're trying to keep it all contained, all inside this building. So on the next slide, you can see our irrigation system. We set it up with, you can see that in the back, there's like a black hose, and each one of these little trees has an outlet for drip irrigation. Although we're starting to move now toward like flooding them with fertilizer water every few days at Dr. Klak's suggestion. Still trying it out to see what makes the trees happiest. But it's really nice to be able to have them on drip irrigation for over the weekend, or if we're going to be out in the field for a long time. It's good to have some backup on the watering. Then on the next slide, you can see our lights there. We have these height adjustable full spectrum LED lights that have really high photon output. We have them on a 16-hour photo period, just like Dakota and Dr. Klak do. And this is our summer intern Hannah modeling our very stylish eye protection that we wear while we're working with the plants. But you can also see in this picture that it's really nice to have the space to work with them. We can really get in there. It's not like a little closet like the highlight growth chambers are, but we can stand in this building. We can move the trees around. We can really see them individually, which has been so useful, so valuable. And then on the next slide, this is a picture. So it's really hard to photograph mesh, it turns out. So this is my dog impersonating a pest. This is our mesh pest curtain. Doesn't keep the pathogens out, but we're trying to keep it as contained as possible because, as Dr. Klak explained, and as we read in many, many places, pests and pathogens are the #1 cause of indoor crop loss. So it's not foolproof, but this pest curtain seems to have been doing a good job so far. We've seen some aphids and some spider mites in our greenhouse. Also have whiteflies this year, but so far, fingers crossed, we have not seen any in the highlight growth building. All right, and then on the next slide, pretty much done. That's our setup. I just wanted to kind of give you a little tour. We're really, really hopeful that we can produce pollen and help Dr. Klak out, because I know it's a lot of pressure to be producing pollen for everybody who's breeding transgenics. We hope that by having a lot of different transgenics, we'll be able to diversify the transgenic lines more efficiently, and hopefully these trees become teenage parents. Who knows, but we're excited. We're really hopeful.
Thank you, Lily. Great presentation. There's a lot of maybes when it comes to science and no guarantees. I mean, this whole organization was built on a dream in 1983. So we're still chasing that dream and getting closer every day. So without further ado, we had a question that why can't they see the questions? Well, there's somebody that's seeing the questions, but I think the folks, the attendees can't see the Q&A questions, but only the panelists. So we'll do our best to get through all of them. We have 14, and we had a couple of good things in the chat that I'd like to address right now. One from Clark Beebe, if I can scroll up to get it. Here we go. Clark said, when I was in high school, we raised and sold certified crown vetch seed. My father always said he wanted the plants to be stressed because they would produce more seed. His idea was fear of death caused the plants to put all their energy into procreation. I think he posted that right after your comments, Dakota, about losing some of the plants. So that was an interesting comment. Thanks, Clark. Hope you're doing well. And then another one. Let's see here. I had it a minute ago. There's lots of little chatter going on in the chat, which is, oh, here we go. From 8 Amish Shield. I have great success spraying compost tea on plants with pests and other naughty visitors. So that's something you scientists might want to try. So with that, I think I'll jump over to the Q&A and start with some of them. There's the first question is off topic, so we'll get to that if we have time. Tim X says, have you tried 24/7 highlight like they use in other hydroponics? Panelists?
Yeah, they die pretty quick.
Okay.
Also, it's it burns out our light bulbs very fast because the high pressure sodiums and metal halides don't have, because they're such high energy output, they don't last as long as you would like, especially when it comes to the LEDs that both Tom and Lily's using down at the TACF pollination shed. Yeah, it's not economical, and also they just really do need some time off.
And I agree, you know, because we're not giving them their actual winter time off at all. We're just treating them like tropical plants, and so and it works. Well, we have success with the 16 hours of light, 8 hours of dark, 12 months out of the year. So that seems to be a good recipe.
There's a question from Mike Alcott. Hi, Mike. Is it possible that the 16-hour stress situation causes the pollen itself to be weird in some way, making the pollen itself less viable?
We don't have any evidence of that. I mean, we test it for the tubes, like you saw in the pictures, and we get viable pollen, and we take it to the field. And last summer we pollinated. We actually produced 5,500 crosses last summer, mostly from our pollen, some pollen from ESF. So we know from the field throughout the native range, from Virginia to Indiana to Maine, and all kinds of places in between, that it works. So I guess that's the evidence that it works, and we are favoring precocious maturation. But that's part of it. Eventually, when we identify exactly what we have in terms of blight tolerant trees that won't die, and we're always, when we're in the field, we're always seeing what the trees look like, on their own. And even the trees that we're pollinating, they're really got cankers and they're horrible looking. And in our GCOs, we've got cankers and pollen and dead trees. So we got to get to a point where our trees aren't dying. And then some will be precocious and some will be tall, slow growing timber trees. And that's cool. We'll have different kinds of results of trees that won't die. We're not too worried at all about the fact that we're kind of biasing right now in terms of precocious maturation.
A related question. Thanks, Tom. If your work is producing pollen in 6 to 8 months, is the pollen sexually mature, or is this comparable to preemie babies?
Yeah, again, kind of related to what the last question. The proof of the pudding's in the eating, right? And we cross it in the field and it works. We got good results in the 40s. Somewhere in the 40s in terms of the percentage that are transgenic, that inherited the gene. We got 47% inherited the gene here in Maine last summer. I think it's a matter of putting the pollen bags on early enough. That's why we're doing it now in Maine. The earlier you put the pollen bags on, the more likely you're not going to get open pollination. So it works. The pollen works. And that's the best result, best evidence that the pollen is totally viable because it does produce fertile offspring.
It's exciting.
And we only collect pollen from specific crosses for that given year. So each year we're outcrossing and then selecting new individuals from new crosses. So if there were any that were somewhat premature pollen, first of all, that wouldn't fertilize the ovule of the female flower. So they wouldn't develop into chestnuts in the first place. And then any archetypes that would be revealed from early pollination should be weeded out by the fact that we select new individuals yearly for this highlight treatment to produce pollen. So all of that should be weeded out with the numbers anyways.
Okay, thanks. John Hempel. Hey, John. I think this has been answered, but are these seedlings making pollen within a year of starting extreme photo period?
Say it again.
Sorry, I said it too quickly. Are these seedlings making pollen within a year of starting the extreme photo period?
Yes. So the one I showed you that we had the breakthrough with here at the University of New England, we sowed it, we planted it in December of 2020, and now we have pollen. So that's a maturation within the same the same growth cycle. So we can now take that pollen out in the field. We have one generation advanced. So that's exciting because we need to diversify. We haven't said this explicitly, but most people probably in this call know we need to diversify away from the original Ellis clone, and this is one of the methods by which we do that.
Cool. Yeah, and then if I had scrolled down far enough, John said, "Oh, I already know that the answer has been this question has been answered," but nothing like clarifying. Some tactical questions. What temperature is frozen pollen stored?
Can be both -20 and -80. We use our -80s just because our -20 is relatively packed at the moment, but it's just fine at a normal freezer temperature if you're using it within the year. I don't know, maybe Tom knows if he has ever used pollen that's been stored longer than the year it was collected before pollination season. I don't think we've ever done it. We've done some viability tests, but there's no point in using 2-year-old pollen if we have fresh or pollen that's within the year.
Right. Yeah, we're going to experiment with that this year because we do have some leftover pollen that's over a year old. And that's where one of the reasons we favor the -80 Celsius, just to hopefully lock it a little bit more in a stable state for a longer time period. We never know, something could go wrong. Lots of stuff goes wrong in science. You talked about your growth chamber breaking. So we want to stockpile it and have it available and have, conservative sort of options. That's where I think the -80 is better. But the other reason is it's got a backup generator. So when the power goes out, unlike a regular refrigerator that has a -20 Celsius temperature, if the power goes out, then you're in trouble. You don't want the temperatures fluctuating all over the place. We know that that's not good, especially if it thaws. You don't want it to thaw. So locking in at the coldest temperature possible is best.
Yeah, there was a related question. Can a household freezer from Fred be used to freeze pollen, or are lower temperatures required? So I think you just addressed that. Okay, I think there's a couple other sort of tactical questions here. I'm trying to group them as they come in. And a reminder, please put the questions in the Q&A, not in the chat, because it's easier to manage them.
Hey, Lisa.
Yeah.
Lisa, this is Jules. Sorry. I'm
No,
just getting a comment from Hannah. I think this is Hannah at ESF. She's saying we used over 1 year old pollen last pollination season using T1 pollen that was produced in 2018. We got offspring from those crosses, so older pollen should work fine too. That's from Hannah at ESF, I believe.
Thanks, Hannah.
Good to know.
Hey, Hannah. Here's a question. Why does the water need to be acidic?
Now, again, I'll send that over to Dakota because he told me to do this. So he said make sure the water is 5.5. I mean, we know that chestnuts like lower pH than like in the 7s, which comes out of our tap at 7 something. But what can you say anything more about that, Dakota?
Well, just American chestnuts, because they prefer acidic soils, they have a hard time uptaking nutrients at higher pHs. So if you're consistently watering at a higher pH, all the fertilizer that you're applying to this tree that is stressed out and trying to produce pollen earlier than it should, doesn't have those nutrient stores. So it's really important that it's able to uptake those liquid fertilizers, the slow releases, whatever you're adding to it, and it won't be able to uptake and utilize those nutrients that you're feeding as well if it's in a higher pH solution. And then over time, you'll just get a chlorosis of the leaves. The leaves will start to yellow, they'll curl, there'll be obvious nutrient deficiencies despite how much you fertilize. So it's just important to keep them at low pHs for nutrient uptake.
Great, thanks. And then some pathogen and pest questions. Robert Gilman, why not do it in laminar flow hoods to keep out the pathogens?
Let me just answer briefly. You know, this could be 100% time allocation to dealing with all the pests and pathogens. It's sort of like, I always feel like we got a lot of balls in the air and we're doing the best we can. And there's all kinds of constraints in terms of the use of space, and different people want the space on campus. And we don't have optimal, constraint of cost is involved. So we kind of do the best we can with what we have and make do. I mean, that's, Lily's solution there at Meadowview is a great one. It's like, okay, what can we do that's really cool within a price, price point that we can afford? So yeah, there's probably other ways to do it in better ways. But we're doing within the constraints we have, we're doing what we can do. Yeah, it's a challenge. The situation is definitely a difficult one. And the key thing is that you've got the plants in there for the long term. So you can never empty that out. Even if you emptied out your chamber, your plants themselves are likely to harbor pests and pathogens. So, that's the conundrum.
And a laminar flow hood is for aseptic technique for molecular stuff. I've never seen a laminar flow hood big enough to hold plants in the first place. And then also you'd have to set up some sort of growth rack in there with highlight treatments. And then if you want to keep out all these pests and pathogens, you'd have to sterilize everything if you're keeping it in a laminar flow hood, which isn't practical. You're not going to sterilize, filter sterilize, or autoclave all of your watering stuff. Fertilizer doesn't come sterile. Soil isn't sterile, and you wouldn't want it to be sterile. There's a lot of considerations, and then you're going in there watering, collecting, all of that stuff. You're not going to be doing that in a sterile aseptic environment, and it's just not practical.
Okay, thanks. Here's a couple for you, Lily. What are, I think Tom covered some of this, but you might have some new ideas. What are some of the methods used to control control pests in Lily's enclosure, and how much was the cost of the building fully equipped?
Yeah, so Tom did cover some of this. Mostly we're trying to contain everything as much as possible. So that was our goal by not, making a drain, because that means putting a hole in the building that things can get into. We keep the door closed anytime we're not working in there. The pest curtain, like you saw, and just keeping a really close eye on them. As soon as we see an aphid, take that plant out, just really, really trying to work these trees every day and keep an eye on them. And then, I'm sorry, I think there was a second part of the question.
How much did it cost?
Was there? Oh, right. How much did it cost? So the quotes we got for the highlight growth chambers that we were looking at were around $40,000, and the final cost of the building was under $20,000. It was about half as expensive.
That's our clever staff at Meadowview. They saved all of your donor dollars with their ingenuity. So well done. Okay.
Yeah, and let me just sort of contextualize that, because the chamber that we use here at UNE, and it's not a very big one at all, to replace it right now would be $46,000. So yeah, so that is very much the case, that you've been very careful with your funds, and you've got, it's really valuable that you can walk around and see everything, as opposed to sticking your head inside the chamber and knocking your head on the top, which I do every single day. Because it's very small. You can keep track of things and watch for pests much more thoroughly than we can. So I love your setup. I think it's very promising.
Here's another one from Jessica. Have you used inoculated biochar in your growing medium?
We have not.
No.
Yeah, I'm not sure what that is. I'd be interested to know.
Maybe throw that in the chat, Jessica, if you would, please.
Yeah, I'm going to write that down. Look it up.
Here's an interesting one from Clark Beebe. It's my understanding that during the carbonaceous period, I probably mispronounced that, when many coal seams were laid down, that atmospheric CO2 was much higher than today. Have there been any attempts to introduce higher CO2 levels in the light chambers to speed growth? Another experiment to try, huh?
We have had talks about that at ESF. Our biggest problem is that our chambers are so old that sealing them is pretty much impossible. So we might be able to do it for a certain amount of time, but we're limping along with how old these growth chambers are, obviously, because we can't afford a new one. And we would have to probably do some serious retrofitting to be able to seal it up in a way that we could consistently keep a higher CO2 concentration. I'm not sure if we've thought about trying that again in the past couple of years, but we've definitely talked about it.
Cool.
Yeah, we don't have the ability to do that either at my university. And again, I'd come back to the fact that our system, as rudimentary as it is, in a lot of ways works. Jared did a calculation the other day, is that we right now have enough pollen available to pollinate 32,000 nuts, create 32,000 nuts. So we have a lot. We're doing as much pollen production as we need for again, everything we're doing is within the strict confines of permitting by USDA. We haven't said that, but that's important. We can only do what we can do within permit restrictions. And so I think we're making tremendous progress on this diversification and production of transgenic nuts within permits. So I think it's working. It's basically working.
Great, thanks. Here's a couple planting questions. In the outplanting picture, what is the 3-inch square black material around the trunks of the saplings? Is it permeable or not?
Good question. Though the ones that we used are not permeable. They come from a company called Plantra that partners with us up here in Maine, and maybe some of the rest of you as well. They used to have those are weed mats and also growth mats. So they do two things: to keep out the competition, which is really valuable, all the weeds that would be wanting to use the same resources as our young seedlings. And then they also warm the soil. So the soil will be warmer underneath that, but still cool within the tube where the seedling is. And we use the tube in order to transition the seedling from the indoor to the outdoor. So the tube is a little bit of a sort of protection device that I think really works well, and we're monitoring the seedlings. They seem to be pretty happy. We put them in the ground about 2 weeks ago. So they are not permeable. The company tried permeable ones in the past, and they switched to non-permeable because they found that these actually hold more moisture, counterintuitively hold more moisture than ones that are permeable that will let more evaporation happen. So that's what we use. When we water, we can water directly into the tube, and then it also then will spread out underneath the mat and stay there much better than if it had perforations. So that's what we're doing, and it seems to work pretty well, as long as you keep the meadow voles from taking up residence, because they also like those mats really well, and they like to live under there and eat the trees. And now we're getting into the category of the pests and pathogens in the field, as opposed to ones that we've talked so much about today in the indoor settings. Lots of challenges.
Lots of variables.
Yeah.
Okay, the questions are coming in fast and furious. Here's a
Great.
couple of homo and heterozygous questions. Are the transgene seedlings homo or heterozygous for the blight gene? And secondly, how can we identify homozygous for the transgene?
So yeah, these are all heterozygotes. So there's only one copy of the oxo gene on one chromosome. So that's why we have to test our individual crosses for inheritance of the transgene. If we wanted to generate a homozygote, we could back cross with a known transgenic female flower. So it would have to be one of our transgenic trees. Ideally, it would probably be a T1 because the T1s are the largest we have in the field at the moment, which we're actually hoping we get some flowers from in the near future, maybe this year or next year. But yes, we would have to cross transgenic pollen onto a known transgenic female flower to generate a homozygote. But right now, our primary goal is maintaining genetic diversity. So outcrossing to mother trees that are not transgenic, that are already established or throughout the range, that is the more important goal at the moment. And also we don't have female flowers that are from transgenic trees.
Just another quick additional benefit of the method we use, which is to produce basically half transgenic offspring, half non-transgenic, full SIB non-transgenic, is that we are producing a tremendous number of pure American offspring. So we're continuing these lines of wild chestnut genetic diversity from throughout the range by our method, because that's what the result is in terms of half. And then now in the field, we can compare their performance side by side, because they're literally in that orchard I showed you the last slide of mine, they're literally growing side by side. So it's exactly the same mother tree, exactly the same pollen, but one inherited the gene, one did not. And we're going to see then over time, over the years, how do they perform? How do they do in the field? And it'll be a really great next step for the science to see how these transgenics do, not only compared to their full siblings, but all the other possibilities, hybrids, F1s, 50/50s, and then also full 100% Chinese. They're all out there. Grand experiment.
Okay, just a process check. We are at 12:39. I assume the panelists can stay a few more minutes. I have to hop off in a little while to return my rental car, so I saved donor dollars by not being charged another day coming from Virginia. So I will turn the host over to Lily, and I think Jules got knocked off even though she's in our office in Asheville. So I'm not sure she's back, but I wanted to point out something in the chat that Jessica shared about biochar. So if you're interested, she put some things in the chat about that. Okay, another sort of pest and pathogen question. Can't you use a soil dredge insecticide systematically or a fungicide soil drench? From T. Morgan.
That's the limitations Tom was talking about with working at a university. It's up to the purview of the lab safety or hazardous waste guys at your respective university. Unfortunately, both Tom and I work at a university where they do not like us using systemic pesticides or soil drenches that kill things, basically. So we're limited to biocontrol, we're limited to hand washing plants, we're limited to much more mundane treatments for these persistent pathogens.
And that's exactly right. It's, I send out questions about what can I do about these fungal gnats, which are just devastating. We're catching them by the tens of thousands on these yellow sticky things, but we still have more. What can we do? And we get some brilliant answers, but we can't use those products inside our classroom buildings. So that's a major constraint and a major challenge for dealing with them. What the answer would be, but then you can't really use it because it's not recommended for indoor usage.
Well, related to that, David Marinelli says, I've had good luck with Bacillus thuringiensis for fungus gnats, mealybugs, and white flies are harder to control without pesticides, although water jets and insecticidal soap helps. Any experience with biocontrols? You might still be.
Yeah, we use not white flies. We use lacewing flies. We do use BT. So we'll mix BT into all of our pots before planting, and we'll do periodic treatments with that. Again, the washing with whatever you just said, the washing through the water jets. The big thing is the limitations of our old building that these chambers are in. We can't retrofit anything to a spigot. We legitimately fill watering cans from a sink and then do our treatments. So pH down to adjust the pH, and then fertilizers and stuff. We just mix that in. So we don't even have like a pressure washer that we could hook up to those. And then also spraying down flowering plants with something that is aerosoled or pressurized would literally wash the pollen away, which is the whole point of these highlight chambers, is to generate pollen. So we try to minimize any agitation of those catkins so we can get as much as possible.
Yeah, those are good answers. Yeah, the water wash is helpful, but again, we don't have the space to do that effectively indoors. You really need to take them outdoors, and that's got its own problems. And again, permitting requires us to keep the plants indoors if they're producing pollen. So we're up against various constraints in terms of pest management.
Here's an interesting pollen question from Gordon, excuse me. Does glass slide pollen collection get all the available pollen? And if not, could it be improved by adding a charge to the plates?
We thought about doing the charge. We actually have a guy on campus who basically, you come with a problem and he'll try to rig something up. He's a mechanical engineer. Adding a charge would be cool, but we haven't figured out a cost-effective and time-sensitive way to do that for each slide, because right now, I don't know how long it takes you, Tom, but when we have 5 or 10 plants flowering, it can take up to an hour to collect pollen, and that's every day. And if we had to do separate slide treatments for each one, just another 2 minutes for each slide, it's a significant period of time. So I'm not really sure. We kind of gave up on that. It might be something that is worth looking into, but at this point, like Tom said, we have so much pollen comparatively to mother trees that are available to pollinate in our permitted orchards, that even producing and collecting the fraction of pollen extra that would possibly be collected on that might just increase our success rate, but it wouldn't increase the number of flowers that we would be able to pollinate because we're limited by the trees that we have.
Yeah, it takes us even longer than that, Dakota. It's a real time consumer to collect pollen, and then all the other, treatments, making sure the plant is happy in all ways, getting rid of the dried leaves and cleaning up, and again, pest management. All of that is more than an hour's worth of work per day. But the slide, what's amazing, again, going back to my slide photo that I mentioned how tiny the pollen is, each slide has hundreds and hundreds of pollen grains on it, right? So just our system that we have right now, we have hundreds and hundreds of slides, and each one is just can produce so many crosses that we're in great shape in terms of quantity right now. Again, the constraint is on the permitting side, the mother tree side. So we're working within that constraint on the pollination side.
This might be a question for Lily, because I know she's been working on this with Jared. Have any crosses been made with transgenic and the best of the B3 F3s?
Yes, we're trying to get as many crosses with the transgenic trees as possible. So we've done B3F3s, we've done pure Americans. Yeah, we're trying to get as much genetic diversity as possible. And we have, I think, 900 some little transgenic seedlings in the greenhouse right now. So we're getting good work done on that front.
Great. Comment on the sticky issue of the slides from Hannah of ESF. Thank you, Lily, by the way. The slightly sticky nature to pollen also helps collect as much possible on the microscope slides along with the static electricity. So as Tom said, they got plenty of pollen to go around and not a lot of trees to pollinate with. So we'll get there. We'll get the happy perfect storm one of these days. From Steve Johnston-Baugh, is Thomas currently using highlight in the open greenhouse setting, or was that an experiment only?
Yeah, I'm only using the plant chamber right now, and we just have 10 seedlings in there. So we cleared out our greenhouse because it's a real vector for pests and pathogens. So we're just going to let it set for a while and figure out what our next moves are. The more places you have your plants, the more things you've got to manage and take care of. And with as much field work as we have going on right now, it's a real relief not to have to deal with greenhouse plants right now. It's very interesting, Dakota, and I learned a lot from both of your presentations. But this idea that you're moving them to the greenhouse after they get to pollen production stage, we have not tried that. And that's a very interesting idea, and it's a cool idea to push them along in the growth chamber and then switch them over to a less intense environment and spread them out a little bit as well. We're going to look into doing that ourselves.
Well, that was circumstantial.
Yeah.
That wasn't by choice.
Yeah.
You can do it, but they won't continue to produce as voraciously, I guess. It's more of, we don't have a growth chamber at the moment. So
yeah, right.
Well, I think that's kind of a fun, happy accident of Chestnut Chats. It's a collaboration opportunity between, three very busy scientists and collaborators. And they're like, oh, I learned something from Dakota. I learned something from Lily. That's really cool. Another great thing about Chestnut Chat. I am going to make Lily host. I don't know if you're comfortable, you three. Can you see the questions? Can you all three see? I think you can go on as long as you want. You're welcome to. We have captured all the questions. We still have 82 folks, or including us, participating. We had over 100 when it started, about 11 questions unanswered. So I hate to end this wonderful discussion, but if you all don't mind just managing the Q&A. I think Jules had some connectivity problems, so she had to get hopped off. I don't know if she's back. There's some good stuff in the chat about biochar, which we're all learning about. But I just wanted to thank all of you before I hop off and turn it over to these great panelists. Thank you. We can't do this without your support. By and large, you heard Lily say, we saved, half as much for the cost of our highlight growth chamber through ingenuity, and we're right in the middle of our spring appeal, and we've had a really successful one. We had a great year last year in spite of all the challenges. So thanks to all of you for your support, your interest, your curiosity. I'm truly grateful for all of you, and I'm especially grateful to the three panelists today. Thanks, guys. And I'm going to get in my car and get to the rental car place before.
Thank you, Lisa.
Thank you.
Okay, I don't know what it means to be host, honestly, but I can see the questions. So I guess I'll just start reading questions. Let's see. Okay, John says, hi, I own 10 acres of mature oak trees. Once transgenic is approved, what would planting them here look like? Clear cut and start from scratch, or prune some of the oaks and wait for the chestnuts to mature?
I would mix them together because the mycorrhizae relationship is similar between the oaks and the chestnuts, and so we rely on oak soil when we plant our chestnuts out as a way to kickstart the really positive mycorrhizal relationships with the fungus in the soil.
Yeah, I wouldn't clear cut. I mean, especially if you have nice oak trees. It would be, chestnuts are both shade tolerant. They're like moderately shade tolerant, so they can grow in a somewhat established canopy. It's more of how fast do you want your chestnuts to mature? What is the purpose of you growing them? If you want them to be more straight and timber type, you know, there's a different way to grow them that way versus if you're just trying to establish them in your oak mature forest. It's really up to you.
Thanks, guys. Okay. Timothy Laurie Clippinger says, please go over your watering recommendations. Seed in pots once sprouted, seedling to 7 to 8 inches once planted outside. And thank you, great work.
Now let's see. So is that, maybe you could go over that again, Lily? Is that for growing seedlings first indoors in preparation for outplanting, or is that for production of pollen? It wasn't clear.
I'm also a little confused. Please go over your watering recommendations. Seed in pots once sprouted, seedling to 7, 8 inches once planted outside. Okay, sounds like the question is about the watering regimen that you use once the trees are planted outside.
Yeah, I mean, chestnuts don't like wet feet. They want, so I think, again, I learned so much from Dakota, you want the soil in general, whether you're indoors or outdoors, to get a little dry, and then you give them water. So it's kind of like going back and forth between a little bit of dry and then substantial water, really indoors and outdoors.
And it's really site specific. And it can be very variable over a specific site. So we really do it based on what's needed. If it's super hot for a while, we'll water every 2 to 3 days, depending on how small they are. If they've been established and have sent out their taproot, they can go a little bit longer without water. That's a really big question because it really depends on site conditions. So sorry.
We do the same. It's very site specific, exactly like they said. We just watch the trees and watch the weather. And when seems like we're going to have a dry week, we water them. If we're going to have a lot of rain, we don't. Okay. T. Morgan asks, how do you address the spiraling root issue in potted plants? Just speaking for myself, we try to up pot them every once in a while. We move them. We start in 3/4 gallon pots, and then before I moved them into the highlight growth chamber, I up potted them into 2 gallon pots. And then in a few months, I'll check the roots. If they need another upgrade, I'll try to find bigger pots.
For pollen production, we actually kind of want that because the more stressed out they are, the more they'll produce pollen. So if we're trying to produce pollen in the highlight growth chamber setting or in a greenhouse setting, we don't repot them. Like I said earlier, we treat them as martyrs. We're not necessarily trying to keep them as healthy as possible. We're trying to keep them in a stressed out level where all they want to do is produce pollen. For trees that we're potting out into the field, they're not in pots long enough to need to repot very often, even at all. We get them out into the shade tents as soon as possible. But this year we had most of our pots in little D10s. So they're like little tubes about this big. And I honestly don't know the sizes of them. I'd have to talk to one of my greenhouse managers, or one of our greenhouse managers, but yeah, it's different depending on what you're growing them for.
For the seedlings that we outplant here in Maine, we use the pots that are 4-inch by 4-inch and then about 14 inches deep. And ideally then when we start growing them indoors around February 1st, and then in May when we're outplanting them, when we take them out of that pot, ideally they've just really kind of filled that whole pot with beautiful complex root structures ready to go. And there's no spiraling problem. I think they're just really ready for that next stage. That's the idea. That's the ideal, that you push them along as well as you can indoors up until May, and then now they're in the field, and we've got really a growth period from now all the way till really November, where they can kind of take their next major step in terms of development, both of roots and roots as well as above the ground.
Thanks. That's really good to know about them needing to be root bound in highlight. I didn't know that, so I'm learning. Thanks.
They don't necessarily have to be root bound, but they will produce faster.
Nice. That's really good to know.
Yeah, we pot them in gallons, and throughout their whole lifespan, we never change them.
Wow. Okay. Thanks.
Sometimes we do check them if they're exhibiting some weird wilting or something like that, to see if there's any like root issues when it comes to pathogens. But just strictly speaking, for root bound stuff, we don't care.
Wow.
And we've started with the 1-gallon pots based on Dakota and ESF's recommendations. We actually use 2-gallon pots now and so for us, it seems like we compared them a little bit. We didn't do anything super scientific about it, but we think they're a little happier in the 2-gallon compared to the 1-gallon. So yeah, not sure which is better.
Interesting. Thanks. All right. Tim Eck asks, will silica gel get pollen dry enough for freezing?
I don't know. We use a dry right, which is like, I can't, it's anhydrous, but I can't remember what what the actual chemical composition is. But if you look up dry right, should be able to find it. And what's nice about the dry right is that it has a color change aspect. So when it's saturated with water, it changes color. So when you have to either bake it out to recharge it to be able to absorb water, or just get new dry right. I'm honestly not sure about silica gel. We've never used it.
Yeah, we used it. You used the dry right desiccant as well for that 24-hour period between collection and freezing. I think it works great.
Awesome. Thank you. Okay, an anonymous attendee asks or says, sorry, as I was interrupted, this may have been answered in the presentations, but what happens to the seedlings after pollen is harvested?
They die, or we try to plant them and they die. They're martyrs. They're so developmentally like messed up after 16 hours of light for anywhere from 6 to over a year, 6 months to over a year, that they're weird. I mean, we've had some survive maybe like a year or two afterwards, but yeah, that's why we also use duplicates of crosses that we have multiple of. We wouldn't put a novel cross into that highlight treatment if that was the only one we had, because they're not going to survive.
Poor little guys. All right. You are conducting this experiment in Maine. What do you expect to be the growing range in the US of the final tree product?
Okay, so what's going to be the suitable habitat geographical range? It
sounds like yes. Yeah.
That's been projected by people at North Carolina State, and it's obviously creeping northward fairly dramatically. So the traditional native range of the chestnut is pretty much the southern half of the state of Maine. I literally got a email this morning from a colleague of mine up in the farthest reaches of Maine, as far north as you go, it's Fort Kent. And he was telling me he's got a couple of germplasm conservation orchards out there that got seedlings from me. And he reports that they're doing great. Some die off inevitably from year to year, but they're either one's 3 years old, one's, in other words, entering its 4th year, one's entering its 3rd year, and they're doing fine up there. So that shift northward is very significant. We kind of look at Maine as being pretty much suitable habitat for the entire state at this point, with the warmer winters and warmer summers. And the problem is certainly on the southern range, where the projections are that it's going to be too hot.
Interesting. Thanks. Okay, Steve Johnsonbaugh asks, 1, how/when do you determine that a tree should be removed from the high light chamber? And yeah, he has a couple questions, so I'll go one at a time.
It either dies or it's produced plenty of pollen and we don't need any more pollen from that cross, but we usually can't leave them in there longer than production season. So anywhere from 6 months to a year, but usually they only produce pollen. I don't know, what is your experience of that, Tom? We usually only get pollen for 2 or 3 months before they're pretty much kaput.
It's amazing that a plant will just suddenly, the leaves will just dry up out of and it's not necessarily sort of, oh, it's being attacked by some pests. Yeah, it just gets to a point where it dies, just very dramatically. So that's kind of, we kind of let them run their own course. And if they're continuing to produce pollen, we will continue to collect it because we want to have that stockpile for the purposes of the future. And as we get more and more places that are allowed to pollinate wild trees, which we have this year, we have more than we had. We sent pollen now for the first time down to Georgia and down to South Carolina. So that's very exciting. So we say any pollen we can collect, we're going to store it at -80 Celsius and it's available for those permitted sites.
Nice. All right. His second question is, do you rotate new seedlings in as trees expire?
Yes. Yep.
All right. Good, simple answer. 3, have you established the minimum light level required to accelerate catkin production?
Dakota,
simple answer is we haven't put a quantifiable number on it. So like par levels, I think they're called. We usually look for a range, but we've never tried to test because we only have one growth chamber. So we can't really have multiple conditions unless we set up different growth chambers. And then there's a whole host of, differing variables that could either induce pollen better or not. We do notice that if the bulbs for our metal halides and high pressure sodiums, if they've been operated for beyond their lifespan, we definitely develop pollen slower. So they're not putting out the same wavelengths necessary to induce catkin and pollen production, which is why we try to replace all of our bulbs every year. That's tends to be, if they're on a 16-hour photo period, it's almost exactly a year for our high pressure sodiums and metal halides before they're out of date. And so, yeah, we basically don't have an actual number.
For a while there, we were measuring PAR. That's photo. What is it? Yeah, photos. Anyway, active range. We were measuring it outdoors, and we were trying to match it up indoors. And so we were doing some of that and getting it sort of matched up. But then I think we moved more in direction, as Dakota's describing, is that we kind of have an idea now of how much they can handle And we know if it's too much, you can see the leaves drying. We try to push them harder. Dakota, you could move your plants higher up if you wanted. You can get them closer to your lights if you wanted to get more light, right? So we can play a little bit around with the distance between, and I know Lily's doing that as well, the distance between the lights and the plants. That also varies, and closer, obviously, the greater the intensity.
Yeah, our ballasts are adjustable, so we could lower them if we want. But the big problem, like you were saying, is burning. And since we can't, because they're developmentally like messed up because of this highlight treatment, some of them grow out in like a wave pattern. Some of them just grow straight up. And obviously the ones that are growing straight up, you could easily char and burn any fresh emerging catkins. And so it's just easier for us to keep them all at the high level and not risk losing any plants that way.
Yeah, we're obviously just starting out, but as I was preparing for this, I read a lot about photosynthetic photon flux density, which is basically just the number of photons that hits a given space in a given time. And like you were saying, it varies a lot depending on how high above your trees you have your lights. We are aiming for 2000 ppfd because I looked up the ppfd for summer in Virginia, and it's around, I think, 1800. So we were going for like just a little bit more than they would naturally get. That said, we've seen a little bit of burning, so then we'll, move them up, move them down if they're not growing, still playing around with it. But that's what we were aiming for, was just a little higher than natural.
Yeah, that 2000 mark is exactly what we shoot for when we were measuring it, and now we have a pretty good idea of what that means just by eyeballing the plants. Yep, that's good. You're on the right track there, Lily.
Thanks. I'm learning so much from this. This is great. All right. LSL-ARM says, how can I make our water more acidic? What should we do so the water works better for the seedlings?
Easy product to purchase. It's called pH down, and it lasts forever. You just put a few drops in your container that you're mixing your water in. So, and you can test it by pH. And for us, we use about a 2-gallon pitcher to water with, and it's 5 drops of pH down, brings it from 7.2 down to 5.5. And we check that regularly to make sure that the water out of the tap hasn't changed, and therefore it would change our calibration.
But so that works for Tom, but it's going to change on like what type of water you have, right? So if you have a higher buffering capacity of your water, if you have a lot of dissolved ions, you might need more for the same amount of water that Tom uses. And then also, just a precaution, pH down is a very strong acid, so you can burn yourself. So just be careful when you're using it. But it's definitely going to be different for each water type. So definitely check it before you add it, because you could either make it way stronger or it might not be doing much at all.
We have extremely high alkalinity water. We have very basic water here because we're on limestone, lots of dissolved solids, lots of all that stuff. So we use sulfuric acid, and we feed quite a lot of battery acid onto these trees, but that's what works for us. So it's very important to know what you're starting with, and then to go from there. We've also used pH down with a little bit of success. All right. Okay. HMCNAB says, Tom, please say a few words about the USDA permitting process.
Yeah, I might turn that over to Dakota, since the permitting process is started and overseen by ESF people, but we make sure that anything we do is absolutely permitted and allowed by USDA APHIS. So our chambers, our outdoor plantings, all of that is approved, and it's constrained. It's we only have 1 acre that we can plant our transgenics out. That's it. So everything has to be within that. So it's very regulated, very strict, and we keep to the rules. It's exactly as laid out by USDA.
Yeah, I'm going to keep this brief just because that is not my specialty. That is my colleague, Andy Newhouse. But basically, we have permitted sites for our transgenics. There are certain rules They have to be fenced in. There have to be reasonable precautions for pests, mainly squirrels or deer or something like that, that might want to eat the chestnuts, that they can't get in and then transfer a chestnut if they were to like not eat it outside and then it were to be planted and grown. If we attempt to do any open pollination studies, we need to make sure that there are no wild chestnuts within 400 meters, which is the effective pollination distance of a wild chestnut. But essentially, each one of the sites for all of our collaborators with the TACF, with Tom, with the Penn State ones, all of that is within a certain permit structure. So everything has to be laid out. Like we have to not tell the government, but we have to, they have to know when we're shipping pollen, where we ship it to. Are we shipping it to a known permitted site? What is the purpose for it? All of those things. It's very, very stringent, like Tom said. But yeah, I'm going to leave it at that because I don't want to say anything incorrect.
Thanks. Sounds good.
Covers it.
All right. Oh, go ahead. Sorry, were you saying?
Oh, that covers it. Yeah, it's it's. I think I continually come back to the fact that we can make a lot of progress, and we are making tremendous progress under the permit constraints. We hope for deregulation some year in the future. In the meantime, we're real busy with the science and making wonderful progress. It's very exciting. And so when we get to that next stage of deregulation, it's going to be a great day, and we'll be well prepared for that, given all we're doing now under the strict permitting.
Nice, thanks. All right, Gordon asks, are your stressed pollen producer plants chosen for specific trait crosses?
Right now, because we're accelerating the process of outcrossing, it's really hard to select for certain traits. So like timber type trees or something along those lines. We have to go off of the basis of the original tree or the mother tree that we're crossing with. But we don't really know what combinations will work well based on their offspring growing, because we're generating these new crosses within each year. So basically our main goal at the moment is increasing to the max genetic diversity, getting all of the odd trees that we find out with all of our citizen scientists. Whenever someone finds a tree, ships us chestnuts, we plant them in one of our permitted sites, and then we try to cross them as soon as they're able to, because right now is when we can save as much of the genetic diversity as possible. Further down the road, once we have more established orchards and we've tracked all the crosses we've made, then we can really start to select for certain genotypes of trees, whether it be timber type, whether it be orchard type, whether it be good nut producers, those types of things. But it's really hard to select for those when we're generating pollen from a tree that's only 9 months old. So really what we're going for is genetic diversity at the moment.
Thank you. All right. Gordon asks, could you start with RO water as opposed to tap water?
We need a lot of water, so we're using several gallons of water every day or every other day. So I guess we could, but practicality is it works fine to just adjust the pH off of the tap water, and the seedlings are totally happy with that simpler method.
Yeah, and I don't know the pH of standard RO water, but I still don't think it would be quite as acidic as a chestnut would prefer, but I'm not sure. We've never used RO water to start with. Like Tom said, it's not practical for greenhouse settings or even growth chamber settings. If we only had a couple of plants and RO water was preferable, I would still check the pH and make sure it's okay. But yeah, I'm not really sure.
Because of our really high pH, we looked into a deionizer for our greenhouse water, and basically, yeah, it comes down to practicality. It's very expensive, and it takes so long for it to process the water that as many gallons as we're putting through, it wouldn't really be doable. But good idea. All right, Steve Johnson asks, what PAR level do you have, Lily? I'm not sure what our PAR level is. I know our PPFD is just above 2000, but I cannot remember our PAR level. I'm sorry about that. Okay, Cindy Custer says, we have 60-year-old forest in upstate New York and fields that we mow 2 times per year. Could I plant chestnuts at the edge of the forest? We have lots of deer. I'd be afraid if I planted saplings that the deer would eat them or they would die for some other reason, and I would feel terrible. Are regular people planting them in fields at the edges of forests, or where, and how protected do they need to be?
Deer will eat them. We, for individual trees, will fence them off, even within fenced-in areas like our permitted sites. I personally, I don't know if you do, Tom, but I personally don't have much experience planting trees outside of permitted locations for us, because that's pretty much all we do. So we have, big deer fences that enclosure our permitted sites. And then we also put secondary fences around each tree, especially for woodchucks, rabbits, those types of pests. I'm honestly not sure, but I can only imagine the more vulnerable they are, the more they're going to get eaten, and especially in a field setting where there's a lot more animals to be hesitant
yeah, the deer will munch them down to the ground. So you got to protect them. You could protect them with tubes. You can protect them with fencing. We're trying more and more to try to protect them with deer repellents, so that, because we kind of like to see them grow nice and wide. So deer repellents is what we're working more on this year. But also the other competition is the weeds and the other plants. So that's why we use those weed guards to try to keep, and we know that from experience that when you just plant a seedling out in without any competition reducer from all the other plants, that seedling's not going to do very well. So because everything else is going to be drawing the resources out of the soil, including the water and the nutrients. So I would say try to absolutely reduce the competition, both from animals, but also from other plants, to give your chestnut seedling the best chance of strong growth.
Next, that covers it well. On the same topic, John Hempel asks, how on earth do you keep squirrels out of anything?
So for our controlled pollinations, we use pollination bags to, first of all, exclude wild type pollen. And then after our pollinations, and we rebag them with these pollination bags, we actually make screen bags with basically your standard average metal window screen to keep out bugs. We basically make envelopes out of them, duct tape the edges, and then just staple the whole bag over top. And then we prune the branches off with a bag attached, and then bring those back to the lab. Tie a cat to each tree to keep the squirrels away. That would be ridiculous, but also pretty cool. But yeah, it's mainly the metal mesh bags that's going to prevent squirrels from getting into your chestnuts. And yeah.
And that's a requirement of, again, back to permitting. You can't let the animals get at the transgenic nuts, and so they're fully protected inside the mesh bag until we clip them down and bring them into the lab and open them up indoors. And it's a lot of work to produce those mesh bags, by the way. I'm so glad we produced hundreds of them last summer, and so we get to use them again this summer. So that was a major task, because it's something that's just not sold. You can buy the pollination bags. We buy them from England. They're $5 each. So they're very pricey, but very cool and useful. And we're using them again. But the mesh bags, you got to make them from scratch. And it's a job in and of itself, but part of the restrictions and what we can and cannot do.
We're ramping up our transgenic pollinations on the farm a lot this year. So we're right in the middle of making 400 of those bags, or the the screen bags. And I'm here to tell you, it
sucks. It sucks.
Yep.
Material.
But at least it'll protect the nuts. That's what's important. All right, we're down to the last couple questions. An anonymous attendee asks, what is the closest college curriculum to Washington, D.C. that includes wood identification? I have no idea.
Yeah, your search would be as good as ours.
Yeah.
No idea, sorry.
Sorry about that. Not much help. And okay, last question. I don't totally understand this, but maybe you guys will. What about CRISP?
Is that referring to CRISPR as a way to insert the wheat gene, possibly, Dakota? What do you think? CRISPR as an alternative to agrobacterium?
Maybe, but seeing as how this is a pollination and growth chamber thing, I was thinking maybe leaf crisp, like the curling and the crisping of the leaves and the highlight treatment. If that's what it's referring to, we just prune off the leaves. Because they're in a highlight setting, because they're under a 16-hour photo period, they're constantly putting out new leaves, and that's just par for the course. So we just prune them off. If it's CRISPR related, we don't have time to get into that. But there have been other talks about CRISPR related to chestnut that you can find online, and a lot of resources. And feel free to reach out to us at the chestnut at ESF email, and we can follow up regarding our experiments with CRISPR if you're interested.
Thanks. All right. That is the end of our questions, I believe, unless, oh, I'm sorry, they just popped up. Jules is saying that we should wrap this up by 1:30. So these are the last couple questions I'm going to answer. After this, we'll consider it closed, I think. But 8 Amish Shield asks, can you use an industrial sewing machine to chain sew? Oh, on the mesh bags, that's worth looking into. Thanks, Jessica. We'll look into it for sure. And then John Hempel says, the mesh bags keep squirrels out of the trans-pollinated flowers, but I gathered that USDA wants squirrels out of transgenic trees too, right?
I don't think so.
That's not preventable.
Yeah, right.
As long as they can't take the chestnuts off of our permitted land, permitted plots, that's the best we can do. If say a branch were to fall off with a bag on it where it could be dragged away, like we check for those and then we remove those bags from the plot and put them in a secure location. But there's no effective way to.
After a windstorm, we go out and we see if anything got torn down, and if something fell down, then we grab it before the squirrels could. Definitely.
All right. So I think that wraps up our questions.
Okay, great.
Thank you so much, everybody who participated. Thank you so much, Tom and Dakota, and Jules for being here. Jules, do you have anything you want to add? Are you back?
No, I'm back. I apologize, but no, great information. My mind is blown, so I'm gonna have to try to sort through all this during lunch. But thanks to everybody.
Thank you.
Appreciate
Thank you.
it.
Have a good day.
Thanks for
Thank you.
listening this long for everybody. Thank you.
Great job, everybody. Thanks, Lily.
Of course. Thank you. Bye.